There you go.
All right. Okay. Yesterday we talked a little bit about how to design micro machines, and the basic sort of design principle that I suggested as a paradigm was working around these critical points in these bifurcations of the energy landscape, or the mechanical equilibrium landscapes. And the idea there was that you could get topological protection, and you could get the lever advantage from working around, not at, but near the critical points. And so today I want to kind of extend that into the realm of second order phase transitions, and show that in second order phase transitions, which really have the same kind of energy landscape splitting, right? In a second order phase transition, you go from a system that has just one energetic minimum, free, if I'm applying the free energy versus some control parameter, okay? As you lower the temperature or something like that, you end up getting this single minimum splitting into two. Okay, that is a critical point, and you can put down the various spinodal and binodal lines. That rings a bell for you, but it's basically the same structure dressed with the statistical mechanics of thermal systems, right? And I want to think a little bit about how to apply this framework, which, you know, started with van der Waals, and Cornell has a very rich history with this problem in terms of the renormalization group approach developed by Ken Wilson, and which won him the Nobel Prize in the early 80s. So I'm going to actually start with the tissue stuff, because that's the problem that I developed for you guys, is a renormalization or a scaling approach for something that's related to the tissues. So we talked about microscopic machines. I'm going to save the viscosity metamaterials for tomorrow, and we'll talk about this grand challenge of printing a 3D organ and what that entails. Okay, so the idea here is that when you take a look at tissues, and so here we're thinking about tissues that are primarily composed of extracellular matrices. So there are two types of tissues that people typically, classify. One are cell-dense tissues. These are sort of similar to kind of emulsions, if you will. The cells are forming a packed structure, kind of like what you saw on the fly wing. The idea there is that the interactions between the cells are the things that are controlling the tissue properties. But there's a lot of tissues that have relatively few cells, and there it's the extracellular matrix that controls the tissue properties. And so here I'm showing you some of the beautiful movies that we've had the pleasure of looking at as we investigate these kinds of things. So it's everything from looking at cartilage of the knee. Over here, you're seeing the surface of the cartilage. Down about 5 pictures would be the bone. And you can see these photo bleached lines kind of tell you what the tissue is doing. So we've looked at knee cartilage. We looked even at jellyfish, tendon, the intervertebral disc, the tissue that's in between your discs is super interesting. It's arranged in lamella, so little layers that are stacked at 45-degree angles. And depending on which way you shear the system, you get very different behaviors. If you shear it in this configuration, then these fibers sort of get stretched as you go in this direction, and then they get collapsed when you go in that direction. The other 45-degree fibers stretch. And if you take a look at them in this direction, you see that most of the deformation happens along the fibers that are pointing towards you, okay, the ones that are coming in here. So you get this very fascinating, like, hierarchical structures that you can generate from these tissues, and we want to start to get an idea of how to design these kinds of things and print them if we're going to make replacement tissues when you blow out your disc, as I am likely to do when my kids ask me to lift them now. It's like they're so heavy. My spine feels like it's about to pop at any second. Okay, that's what happens when you get old. All right. So I'll tell you the story that we investigated for articular cartilage, and we'll see how we go from there. So articular cartilage is the soft tissue that coats the long bones in your body. Okay, so we're talking about like elbow, we're talking about knees. And just to give you some background, arthritis, which is what happens when this tissue gets degraded, is a pretty painful disease. It causes joint mobility and discomfort, and about 46 million Americans will suffer from this disease. In fact, like almost a quarter of all the visits to your primary care physicians are going to be based on joint pain, right? So it's an amazing material in that it lasts 60 years or so without like any issues, really, millions of loading cycles each year. But our ancestors were not designed to live that long. And so as our age creeps up, our average age creeps up, those of us over that age tend to develop these kind of joint pains. And so it's a pretty important problem, and it really has a large cost in terms of the effect on the economy. You can even tabulate that. Okay, so what do we know about articular cartilage? A lot of this comes actually from a paper that was published almost 100 years ago. This guy Benninghoff like meticulously looked at cartilage and was able to sort of deduce this arcade structure. So if you take a look over here, this is the diagrams of Benninghoff. And what he saw was that the fibers in cartilage were arranged in these arches. And the fibers turned out to be collagen fibers. Near the surface were arranged in these kind of layers. They were parallel to the surface. Near the bone, they were perpendicular to the bone. And in the middle, they were kind of transitioning. And what's amazing is that, in 2011, when we can start to do EM microscopy, cryo-EM microscopy, that structure pretty much turned out to be, bang on, right? We have this tangential region where the fibers are nearly parallel to the surface. We have the deep zone where everything is radial, perpendicular to the bone. This is calcified cartilage. This is where the bone is. And then in the middle, you have this kind of messy stuff that, where the transition happens, right? And one of the big questions that we've had is really trying to understand how the mechanical function of this tissue arises from this structure. So one thing that is really interesting and kind of curious is that it doesn't matter what animal you take, everything from a mouse, and you can do this in horses and elephants too, all of these animals have different thicknesses of cartilage. Okay, so a human has maybe 5 millimeters of cartilage coating their bone, but a mouse might have even less than a millimeter. But in all of these animals, the part that's preserved is the surface region, right? So that region is the same in all the animals, but how thick the deep zone is can vary quite a bit depending on what animal you are. So we wanted to kind of try to think a little bit about why this is and how to understand why this region is preserved and what its function is and how the properties emerge from the structure of the cartilage itself. So here's kind of like what was known. People had measured the full thickness properties of cartilage. People have also done things like take the cartilage and cut it into zones, like you just lop off the top part and measure the mechanical properties of that. And people had even sort of taken a look at cartilage using videos, but it wasn't particularly good because they were looking at cartilage without many cells and they couldn't track anything. And so we thought that we could do better. And so we, like our previous speaker, developed an apparatus that allows us to load the cartilage between two plates and then apply a shear to the cartilage, and then we could image it from below. right? So this is a tissue deformation imaging stage. And the thing that's nice about this particular device is that you have a little bath that you can put PBS in, just some buffer to maintain the cartilage properties, so you don't want it to dry out or anything like that. And then you can image below with a confocal microscope, and you can even paint lines to tell you how those lines are deforming. So if you photo bleach the lines into the cartilage, you can actually see how the tissue is deforming, right? Then you move one of the plates back and forth. You measure the forces on the back plate, the one that you're not moving. And if the area is conserved, which it is, then you can measure the stress as a function. The stress is uniform throughout the tissue. And so if I'm looking at the deformation in the lines, you can back out the modulus as a function of depth. How am I doing? So far, so good? It's clear? All right. Okay, so here we go. From now on, I'll put the tissue surface on the left, and the deep zone will be like 5, pictures. The bone will be 5 of these kinds of images to the right, right? And what we see is that if we paint just vertical lines, or in this case horizontal lines, when we shear the tissue, those lines are not just moving diagonally in parallel. You're getting these S shapes. What does that mean? That means that the tissue is more compliant in some of these regions, right? The place where these slopes are highest are telling you where the tissue is straining the most. And if the stress is the same, it's telling you that in this region, the modulus is more compliant than in another region. If it was infinitely stiff, those lines wouldn't bend at all, right? They would only shear in the weakest regions, right? And so that's what's going on here. The tissue is very stiff down here. It's more compliant over here. That's why it strains more. And then it's a little bit stiffer near the surface. Yeah? Questions? Yeah. Yeah, so it turns out that it doesn't much matter. Like we've done this with human tissue, we've done it with horse, we've done it with bovine, and we are using bovine here. Okay. Good. Yeah.
Part is different for animals. Is that starting after the thick part, like the stiffer part at the top? Is it after the stiff and the?
Yeah. So this is all the surface region. Okay. Over here, each of these cells, you can see in the cartilage there are lacuna, which are these like holes. The cells are in the lacuna. Each cell is about 20 microns, and so the whole region over here is the surface, and then the deeper zone is much further down. So this is the part that's preserved in all the animals. Good question. All right. So it turns out that you can look at the modulus and you can measure that it has a difference near the surface. So here is the average modulus as a function of depth. And what you're seeing is that the modulus near the surface is about an order of magnitude more compliant than the deep zone. Okay, so it can really vary by a lot. An order of magnitude is not a small amount. It's not 10% or anything like that. It's really a factor of 10 more compliant near the surface than it is near the deep zone. Right? So we can do things like we can measure the mechanics, the stress that it takes to deform the system at different depths. And here's what we find. If we do this at very deep regions, we get the red curve. This just tells you that as I strain the tissue, I'm getting a Hookean response. All right, this is just a spring. But if I do this near the surface, I get this blue curve. And that's telling me that it's not just elastic, that I have a viscoelastic response. The fact that it's thick is telling me that there's a viscous dissipation that's happening in each cycle. This is kind of like your work cycle in thermodynamics, right? The area under this curve in a PV diagram or a stress strain diagram tells you how much energy is being dissipated. And so what we find is that when you take a look at cartilage, almost all of the energy is dissipated by this region over here near the surface. Okay, so everyone thinks of cartilage as a shock absorber, but under shear, all of that shock absorption is happening near the surface. Okay, it's not evenly distributed. 90% of the energy is being dissipated near the surface. Yeah.
How you measure it? Like, how do you get a local?
Yeah, what you do is you take the displacement. The derivative of the displacement is the strain. And then you have a stress that you measure. You move one of the plates, you measure the stress over here. So a stress divided by a strain gives you the modulus.
Right, but that's usually like, I have some stress, which is already a derivative.
The stress?
Stress is, well, okay, you have some stress, which is the
You're applying a force per area. Yeah.
And then ratio between that and the
Strain.
derivative of the displacement.
Yeah.
There's different directions. So
no, but yeah. So this tissue is pretty much a one. So it's pretty homogeneous in this direction. It's layered, and so we're really only looking as a function of depth. So if you take a look, it's pretty homogeneous laterally. So you're really looking at the derivative as a function of depth.
Depth as in Z?
Z, yeah. So in this case, Z is in this direction.
So the
0
strain
to.
is a derivative in, let's say, X to Y directions.
It's this slope, the slope of these lines. Okay, so I have these lines. They have a certain slope. Okay, so I can measure
That's
the change.
strain.
That's the strain. Yes.
So that, okay, so but that's the modulus, but now you want it as a function of Z.
Yeah. So for each layer, I have a strain. Okay, so let's do this. So I have the displacement as a function of Z, and it might look something like this. I have the strain, which is the derivative of this. The strain might be, quite low here, and then it has a peak, and then it goes, really low, something like that. Okay, and so that's the strain. And then you get the modulus, which is kind of like the inverse of this, which is sort of how you get these.
I think the implicit thing is that simple shear, the stress is the same. All.
Yes.
That's the thing you're not saying.
Right.
Sorry, the stress is, the area is not changing, so the stress is the same. That's Newton's second law.
It's measured across that whole.
It doesn't matter. So that the force has to be, if I'm applying, here's my tissue, here's what I'm measuring. I'm measuring this is a spring. Okay, it's like a plate, but it can deform in two directions. So if I'm moving this back and forth, those forces get transmitted to here. The forces are balanced the whole way through. Okay. Correct. Correct. So you get the strain versus Z, and then you invert that to get the modulus versus Z. And you find that the modulus is more compliant near the surface, which is the top curve. And then from looking at the Lissajous curve, the stress-strain cycle in oscillatory shear, you can back out the energy dissipation. Yeah, exactly. So that's what this says. You have a huge imaginary part near the surface. That's where all the energy is being dissipated. Okay? And that's important because if you take a look at medical procedures, in the past, people used to think that cartilage is cartilage is cartilage. So if you damage the surface of your cartilage, what you do is you go in with a little Dremel tool and you shave the cartilage, okay, to provide you with a nice, clean, pristine surface. This is an example of what happens when you do that in rabbit. So over here is the cartilage after it's been debrided. And you see all these little dots here, okay? Those dots are cells. And if you look in the bottom image, which is just a couple of weeks later, there are no cells left. Okay, so when you get rid of that energy absorbing layer, bad things happen. Okay. All right, so that's one of the important ideas here is that these surface regions can actually be really important for the tissue function. And we'll talk more about that towards the end of the lecture. Okay, so what I really now want to ask about is how does this property emerge? Yeah. Yeah, it's a little bit different. I mean, what happens is that when you're young, the cells are everywhere. The cells near the surface are a little bit squished compared to the cells in the deep zone. But as you grow older, the tissue becomes acellular. So like my tissue probably has very little cells left in it. And then you're just relying on, you know, the few cells that are left to kind of maintain its homeostasis. Yeah.
Relate the, you know, your geometry of Z and surface versus not surface.
Surface of one bone and Z, here's the bone, or surface of another bone, this is the femur and the tibia. So in the femur, you go Z in this direction.
So meaning lower is where the
This is stiff. This is stiff. This is the compliant region, and the cells are, you know, when you're starting off as a newborn, you have cells everywhere, and then they kind of degrade with time. Meniscus is different. This is the meniscus, and usually there. So because we care about these things as older individuals, let me take a moment to describe some of these problems. Okay, so first of all, I am pointing at you. So first of all, the meniscus is like a crescent-shaped thing where the condyles sit on, okay? And so that prevents the two bones from getting like really large. So sometimes you get tears in the meniscus, those get lodged, and now your joint has a harder time moving. And so what you need to debride that. There's nothing you can do. You have to kind of chop that stuff off. Okay, but in the case of the femur, right, you have these two condyles sitting on the tibial plateau, and those are sort of sliding. That's the death that I'm talking about when you debride it, okay? Yeah, and you might say, okay, well, why not just get a knee replacement? Knee replacements are great. The technology is really improved. But knee replacements, the way it works is that you lop off this part and you jam a little platinum sort of piece into the femur. Okay, so now you have like a bionic joint. Awesome, except for that it seems like the pieces of the metal get sloughed off in time. And after a while, they get lodged in where you kind of glued the joint into the bone. And so after about 10 to 15 years of function, it has to be replaced. But the problem is that your bone has been degraded so badly that there's nothing to stick the new implant into, okay? So you're done. Like after 15 years of a knee implant, especially if you use it like vigorously, it's really hard to repair a joint. So you're immobile after that. So that's why we push everybody to get the knee replacements as late as possible with the idea that you get the knee replacements, you're less mobile when you're older, and then eventually you die before you need a new knee replacement. Okay, those are the hard facts. All right, let's talk about physics, not medicine. So I want to understand a little bit about this structure and fiber organization and how it contributes to the tissue function. All right, and so the paradigm is that existed in the field was that it's this orientation of the fibers that's really giving rise to the mechanics of the tissue. That near the surface, the fibers are horizontal. Maybe that creates a more resistance to shear. Near the bone, it's vertical. You know, how does that, you know, affect compression, things like that. And so I should say that the compressive properties of cartilage have been kind of figured out for a long time. Cartilage is a poroelastic material, which means that it has like a fiber network and water inside. And so if you compress it very rapidly, the water can't squeeze out. And so it becomes very, very rigid. If you press it slowly, the water can leak out and the whole thing can compress by like 75%. You can really smush down cartilage, all right? But the shear properties, nobody knew about, and this was the paradigm that people had thought about, that near the surface, you have this collagen organization that's different, so that must lead to a different in mechanical properties. And we know that from our measurements that the surface is indeed more compliant. So there's a precedent to trying to figure this out. Yeah. These are cells that they secrete both collagen and the secondary component of the extracellular matrix are called proteoglycans. So the collagen are pretty stiff fibers, and they're usually not just like single collagen, triple helix, they're bundled together into fibrils. And so you think about like a polydisperse sort of network of fibers that are between like, tens to hundreds of nanometers in diameter, okay, by the time they're bundled together. And the proteoglycans are essentially little sugar, they're like brush-like molecules attached to something called hyaluronic acid. So they're like brush-like molecules. They're highly charged, which means that they attract water through osmotic swelling. Okay, they osmotically attract water, which screens out that charge. Yeah, we'll talk a little bit about that, at least as best I understand it. But there's certainly going to be inhomogeneity based on where the cells are at. Okay, and it's a super interesting problem how the cells know what to do. Like they're in these lacunas, they're attached to the lacunas with little fibers, and so they're getting mechanotransductive signals that tell them which elements to produce, right? Should I degrade the collagen? Should I increase the aggregate? Things like that. Okay. Yeah, they don't. It's avascular, which means that nutrients have to diffuse in there, which also means that the cells are very hardy. They're like, Al Grzinski used to say, they're like cockroaches. You can't kill them. They're amazing. Like you can do anything you want to this tissue and the cells are great. So it's really easy to work with, relatively speaking. All right. So what we did was we started measuring the polarization of the fibers using polarized light microscopy. You take a thin section of the tissue, you put it between cross polarizers. If the tissue has any polarization, then it'll rotate the light. And so if you have cross polarizers that are going to block that light, if you put the tissue, if the tissue polarization is some angle to that, then you'll get light through. And indeed, when you take a look at the tissue, you can see polarization of the fibers, especially near the surface where it's really, really polarized. You can also do things like quantitative light microscopy. You can also quantify the orientation of that polarization. And when we look, again, it looks very promising. So if you take a look over here, this part is highly polarized. It has a slightly different angle depending on where you are in the medium. Okay, so here it's really parallel to the surface. Here it's more random. But the problem was that the length scales didn't match up. Okay, so this polarization only occurs in the first 100 microns. And that soft region of the tissue really extends the first 500 microns. And so when we did a correlation analysis to look at the modulus as a function of the polarization index or the angle, it was kind of a mess. Like we just couldn't see anything really great. Okay, so that means that this paradigm that had been, really in play for almost 100 years needed some reworking. Okay, at least for the linear mechanical properties, which is what we're measuring when we're doing these experiments on the cartilage tissue. Okay, so far so good.
It looked like there was this very large region of 500 microns that was softer, but maybe there was something a little bit stiffer near the surface.
Yep. It doesn't look like it correlates for the linear response. Yeah.
I feel like there is this correlation depth and modulus
Yes.
and orientation and depth.
Yep.
But there's
Yeah, because they're not aligned. Okay, so if you take a look at the stiffness of the tissue, right, the stiffness of the tissue is not like lining up perfectly with the orientation. Okay, so that means that you don't, like when you look at these, they look kind of scattered, right? At least there's nothing strong that you're seeing here. Now, maybe there's a subset of the data that you could squint your eyes at, but it doesn't look great. Okay, so let me tell you a little story. I think Mumita Das, if you were here for her presentation earlier, told you a little bit of this. I'll tell it to you from my perspective, and then we can sort of go from there. All right. So the idea is the following. So here's our cartilage tissue again. Here's the surface. Here's the bone. As I said, you have these very stiff collagen fibrils and fibers that are permeating the tissue. Here are the aggrecan chains, and here's the hyaluronic acid chain that's binding them all together. And so the idea is that in shear, there really was no existing framework to understand the data that we were measuring. Okay, so the question is, how do you build such a framework? And the way you do it is you try to correlate the constituent properties, right? Like what the thing is made of with the modulus. So in particular, you could measure the cartilage composition. You might see that there's some relationship between the aggrecan and the collagen, depending on the depth or something like that. But either way, you can try to look for different points in the tissue where you have some amount of aggrecan, some amount of collagen. You might do something to then measure the associated shear modulus. So you would do an confocal elastography measurement, like I just showed you, to then determine what the modulus is that's associated with that given concentration of the constituents. So far, so good? All right. Another thing you could do is you could degrade one of the components. So for example, you could use trypsin to degrade the aggrecan molecules. Now you would get no aggrecan, and only the collagen would vary along this axis. And then you could measure that modulus as a function of collagen concentration only. And then you'd have to figure out if there's some sort of model that can fit these curves and give you predictions for other places in this phase diagram. Does the protocol, is the idea clear? So far, so good? How do we measure it? Yeah, let's do that. So let's see if we can actually do this. All right. Okay, so like I said, we get our tissue from the local butcher. Here's Moshe giving us one of the legs. Actually, this is probably a pig leg. Anyway, we get cow legs, so it's kosher. We then dissect that leg, and all the meat goes to the students. So if you want to eat steak every night, you want to work in this field. You then dissect out the cartilage, and you use a little punch to take out circular punches. And then if we want to do the trypsin degraded or healthy cartilage, we can either add the digestion of the cartilage or not. We then bisect the samples. Half of them go to our FTIRI, which is how we're going to determine the concentration of everything, and half of it goes to our elastography measurement. All right, so they're matched samples. So I know for this elastography measurement, I'm going to know exactly what the depth-dependent concentrations are for the aggrecan and collagen molecules. Yeah. So it's as good as we can. You're right. So if I have sections, I may not be getting the average depth, but it's as good as we can get it. Good questions. All right, so let me show you what this looks like. So here's a little section of tissue where we're doing histology, which means that we're just painting the aggrecan molecules in here. And if we have healthy tissue, you can see that a lot of it is red. That's telling you there's proteoglycans all along the tissue. If you then look as a function of depth with Fourier transform infrared microscopy, let's say we look at this little section over here, we can measure the profile, okay, the absorbance as a function of wave number. And what we can do then is we can also do that measurement for just a vial full of collagen or a vial full of aggregate. And then we can add those two in order to try to fit the data, and this is what it looks like. So the fact that you're seeing very little difference between the measurement that we have for the cartilage and the sum of these two things tells you that we can fit the experimental data and extract the concentrations of aggregate and collagen. And you can do this for healthy tissue. You can also do it for degraded tissue. In our degradation measurement, we kind of protect the sides of the tissue, so the degradation proceeds as a front from the surface on down. And in this case, you'd measure 0 aggregate absorbance, and you'd get only collagen in this region, and that predicts the data very well. Okay, and so that's how we back out the concentrations of aggregate and collagen across the tissue in the degraded and in the healthy regimes. Yeah. What does that mean they're not interacting? Well, they are interacting in the sense that the aggregates are interlacing in between the collagen. But the idea is that if you mean for the degraded one, like when you're degrading, you can really specifically degrade just the aggregate. This is measuring the bonds in the actual molecules. So it's not really sensitive to any, this is really bonds in the molecule. So you can, there are certain peaks that are related to bonds in the aggregate. There are certain peaks that are related to bonds in the collagen. So you can really differentiate these pretty easily. Okay? Great questions. All right, so that allows us to get the collagen concentration as a function of depth, and the aggregate concentration is a function of depth. And we can start to do this plot, where we now have, for the blue curve, which is the healthy tissue, we have the collagen concentration and the aggregate concentration. That's this data. We can also get the aggregate concentration and the degraded region for the aggregate concentration, and that's the red data over here. Okay, sorry, collagen and aggregate. Okay, so that's the red data. So in this regime over here, down to about 1 millimeter, you do not get any aggregate. It's 0. So that's what this data shows you. All right? So far, so good? All right. So what about the elastography? So we talked about that a little bit. You paint these lines across the tissue. One thing that we had to do, which was pretty painstaking, is you don't want to compress the tissue. You really want to have it be uncompressed. So we had to figure out how to glue the tissue very, very lightly so that you're not like getting the glue into the tissue, so that you can shear without compressing it or extending it. All right. So that was difficult to do. But we figured it out. And then we can measure that shear modulus as a function of depth. And you can see differences between the healthy tissue in blue and the degraded tissue in red. And you can then look at the modulus as a function of, so if you have the aggregate and collagen concentrations, you can look at the modulus as a function of the collagen concentrations for the two different conditions. And that's what gives us these curves over here. And the thing that I want you to really notice is that this is quite amazing. This modulus here can vary by almost 2 orders of magnitude. But the concentration of the collagen is only varying by less than a factor of 2. Right? And so when you change the concentration of something by a very little bit, and you get a very large change in the other properties, that's telling you that you're near some sort of mechanical phase transition. Right? And so that's what we want to kind of understand is, can we start to develop a model that explains these very large variations in modulus with very, relatively moderate variations in the concentration? So I want to give a shout out to really pioneers in this field of trying to apply phase transitions to biological systems. So I was super inspired by the work by Sarah Keller and Sarah Veach, where they showed that lipids on the surfaces of cells are sitting next to a second order phase transition where the lipids form different concentrations in one phase versus the other. And they showed that basically when you take these two lipids and you look at their concentrations, they're sitting next to a second order phase transition where they want to separate. And what the other 13 or so lipids on the membrane are doing is maintaining the distance of the primary lipids from the critical point. So again, it's that same kind of philosophy of keeping yourself near but not at a critical point in order to be able to determine and control the phase separation. And in this case, these were important for things called rafts on the lipid membranes of cells. Okay, other folks that are doing things like this, Cliff Brenwin has been showing that you can get liquid phase separations. These are called P granules in cells. And this has become like a major area of study. Eric is super interested in this, as are many, many people. And we're going to try to look not at like a phase separation, but rather something called rigidity percolation, which was something that was developed by many people, but where I learned a lot of this from was from Fred McIntosh's work and from Mumita Das, right? And the basic idea behind rigidity percolation is that when you're forming fiber networks or networks like these on lattices, the modulus, when the lattice is not percolated, the modulus is 0. And once you go above the rigidity percolation transition, your modulus is finite. And so for a very small change in concentration, you can get orders of magnitude change, because from 0 to finite is infinite orders of magnitude change in your modulus. All right? And it's a little bit complicated. There are different things that you can put into these rigidity percolation models. So for example, when a network is just percolated, then you have a very tenuous set of bonds that are connecting the two sides of the tissue. If you care about angles between these bonds, if you penalize those energies, then the minute you have a percolated network, then you have rigidity. But if you don't care about those angles, right, if you don't care about penalizing these and you only care about maybe stretching or bending of the fibers themselves, then instead of just percolation, you can get to something called just rigidity percolation. And this is the underlying phase transition that is thought to really govern the properties of these networks as they pass through and become denser. Okay? So the idea is that if you have rigidity percolation, there's some concentration rho. If you look at the modulus, where this thing kind of shoots up like a bat out of hell. But if you have things like bending that are much weaker than the stretching forces, then this thing sort of gets smoothed out, sort of what's called an avoided singularity, and you sort of smooth out this transition over here. And if bending forces are really important, then you might have some kind of, double, step or something like that. But the idea is that this stretching transition is the underlying thing that's really governing the mechanics of these rigidly percolating systems. Okay? Is that clear, Leo?
Latter case where you have bending before still be kept as a critical phenomenon. Looked at that critical point.
It's actually difficult. Okay, people have looked at the stretching one. So if you don't penalize bending, then it's a real critical point. And then if you have bending, like if this, then it's sort of like an avoided critical point. There's another one over here at percolation, okay? And there's a lot of stuff. Yeah, that's just geometrical percolation. And so we'll talk a little bit about this. I want to talk a little bit about this. Yeah.
Describing operating near a critical point as something dynamic, right? You're taking very little your concentration and isn't that?
I haven't described the advantages yet. I'm just saying that the system looks like it's operating near a critical point. but I'd like to tell you about the advantage.
Seems to say that it would be this at
I understand what you're saying. So let me, if you're not satisfied in a few slides, then we can chat more about it. But you're absolutely on the right track, right? Like, why would the tissue want to do this? Great. So far, so good. Okay, so let me sort of describe just the rigidity percolation part of things. So as Umita probably showed you, you have, she models these systems using a 2D Kagome network. Kagome networks are nice because they only have 2 fibers crossing. She does use the bending moduli of these things, so there is that part of it, but the idea is still the same. If you have a low concentration of these bonds, you're floppy. As you start to become more filled, as P increases, the probability density of bond occupation increases, you start to develop rigid clusters that can transfer forces, right? And then as you fill it even more, that network becomes stiffer, much, much stiffer, and eventually the thing saturates as you get to almost full bonds. So when we think about our phase diagram here, our phase transition, our mechanical phase transition in cartilage, the idea is that this region down here is still rigid, but it's barely above the rigidity percolation threshold. Whereas this region up here is much more dense, okay, near where the collagen concentration is much higher, near the deep zone of the tissue, it's much more dense. That's why the modulus is so high. And by working near this critical point, if I erase the confounding variables, just get the moral picture across, by working near this critical point, if I'm down here, changing my bond occupation probability by just a little bit moves me a long way up this curve in terms of the rigidity. Okay? And I think here I'm plotting it log, and here I may not, or here I'm plotting it log, and here I'm plotting it log ln or something, but it's the same idea.
for degradation of this, that's really bad.
Hold on, we'll get there. Okay, now if I have the healthy cartilage, okay, so I can actually put together a model that fits that. If I look at the healthy cartilage, the idea is that, again, you have the same property, but now you have to account for the aggrecan molecules. And the way that Momita modeled that is by assuming that the aggrecan molecules form a gel that's sort of underlying the network. Now, if the stiffness of that gel is the same as the stiffness of the collagen network, then the whole thing is rigidly percolated. But if it's somewhere between 0 and the collagen network, then the stiffer that gel, okay, the higher the concentration of the aggregans, the stiffer that gel, the more effective connections you have between the collagen fibers. So far, so good? All right, and so the idea then is that she was able to model the entire surface pinned by the experimental data along these 2 curves. And one thing that was also satisfying is that when we compared the numbers, the predictions that her model had for the shear modulus for a system that's very low in collagen concentration, but quite high in aggrecan concentration, so like an aggrecan gel, she was able to nail this modulus bang on, right? So again, it gives you some kind of picture. And this is a very interesting picture because what it says is that, you know, this very non-trivial surface can really kind of guide you to try to understand the mechanical properties, the linear mechanical properties of cartilage. Like, for example, the fact that this takes a steep nosedive over here means that there's a highly nonlinear relationship, okay, between loss of aggrecan and loss of collagen, depending on where you are, right? If I'm deep in the deep zone and I lose a little bit of aggrecan, you know, my modulus is going to decrease by a factor of 2 across here. But if I'm near the surface and I lose aggrecan, I can get, you know, a very large factor. And so as tissues start to degrade during diseases, right? You might use a diagram like this to kind of plot the trajectory of diseases along different locations in the tissue to try to understand whether or not there's like a tipping point for arthritis when it develops in cartilage. Yeah. Yeah, I mean, there are a lot of assumptions that go into this, okay? So when you're thinking about cartilage properties, you're really thinking about collagen. It's not just a single fiber width, right? It's polydisperse. You're thinking about the aggregate concentrations. You're thinking about the hyaluronic acid, collagen concentration, collagen cross-linking, collagen connectivity. All of that is kind of condensed into these 5 model parameters. And we did our best when we could to constrain these parameters from the data in the literature. Okay, so it's not bad. And let me kind of give you our sort of why this model is helpful in creating a framework to think about now the advantages of doing it this way, right? Okay. And for details on that model, you can kind of look at this set of papers down here. Okay, so why is this an interesting way to do things? What's interesting is that in cartilage, the collagen network, okay, takes months to years to reorganize, right? So if you damage your collagen network, it won't be for months to years before the cells can produce enough collagen in order to restructure that network. But what the system does is it puts itself right near the rigidity percolation threshold. And that means that by altering the aggrecan molecules, which happens on a timescale of days, I can rapidly shift my modulus up and down near the surface. Okay, so what we think is going on is that cartilage is behaving like an active tissue that is receiving mechanotransductive signals about the usage of the cartilage and then adjusts the behavior, the modulus locally by producing or degrading the proteoglycan concentrations in order to kind of move you up and down this very sensitive modulus curve. Okay, so we think that's what's going on, and that's why this is an advantage. Right? It's a way of actively controlling your modulus when you have a primary network that's really bearing most of the load that can't be turned over very rapidly. Clear? Yeah. Yeah, so we're trying to develop techniques for measuring the biology of the cell's response, the mechanotransductive response to tissue deformation. And it's complicated. Each tissue has 5,000 cells, and you're trying to do a deformation and then see what the response of the cells are. But there's lots of mechanotransduction pathways that tell the cells whether or not they should start, the death cycle, apoptosis, or if they should produce more aggrecan, produce more collagen. And we're trying to now figure out how to do this en masse at the tissue scale. And it requires some machine learning algorithms so that we don't have to constantly track the cells and do, anyway, it's a major effort, but it's something that we're interested in doing. And actually we just got funded to do it. So next Boulder Summer School, talk about that. Yeah.
Characterizing the collagen network.
Yeah.
So then I should think of it as the curve is shifting as a function of a PLF.
Yeah, so that's one way to think about it, is that the aggrecan is shifting the location of the onset. Yeah. Okay, so now I want to talk about how that kind of framework, okay? So one thing that you can do with the collagen network, right, that aggrecan network is swelling the collagen fibers. If you swell the network, then you go from this blue curve, which has a rigidity percolation here, to a swollen network. Now, if I have any bonds that were not stretched before, when I swell the thing, those bonds are stretched. So that means that you're moving to a new rigidity percolation curve associated with a pre-stressed network. That's stuff that Andrea was talking about yesterday. So you're imagining that you started off over here. If I produce more proteoglycans, I'm shifting that whole curve over. And therefore, that concentration now is well into the percolated regime. Exactly. Okay, so there's a beautiful mechanism for essentially shifting these transitions over. And once you have the power to shift these transitions over, you're really in business in terms of massively controlling the properties of your tissue, right? So this, I think, is another like point, which is if you can control the mechanical phase transition location, right, with this secondary network, you can do a lot to control your mechanical properties, right? Is this concept clear? Okay, so now that's all isotropic. I haven't even gotten to the anisotropic part yet. Yeah. The numbers are from simulation. Don't pay attention to the numbers. Okay, the concept is what I want to get across. The numbers in anything that we publish experimentally are to be believed, but the numbers in the simulations, I'm just giving you an idea. Okay. All right. I can also do things like I can swell the tissue isotropically, but then I can compress the tissue uniaxially. Okay. So what does that do? That takes you from this red curve that you see over here. When I compress the tissue uniaxially, I go to the green curve that's over here, right? And there's a difference, right? Because now it's anisotropic. If I swell everything isotropically, fibers are being pulled in all directions. If I compress uniaxially, only the component of the stress tensor in the axial compression gets modified, right? I can then do compression and shear. If I shear the fibers, okay, then I'm stretching them again. So I might have compressed them here, they buckle, they're no longer contributing to the modulus, but now I strain them under shear, they get stretched. So now they are contributing to the modulus. That's what's happening in this yellow curve over here. And so when you're really thinking about the function of the tissue, you need to understand how it's playing with these mechanical phase transitions to really understand why it is that it can last 60 years, which is, by the way, better than any man-made material that we can fabricate. Millions of loading cycles, 60 years, we don't know how to make that. And our conjecture is that it's by learning how to manipulate the properties and adjust them based on the proximities to these phase transitions that the tissue is able to survive that long. All right, so again, the idea is that if you're in this pre-stressed state, the minute I compress the tissue, now the curve moves over and I go down in my modulus. Then I shear the tissue. What's happening there is that any bonds that were not stretched before under shear will get stretched. And now I'm essentially moving from this compressed tissue, uniaxially compressed tissue, to now a stretched along the shear direction part. Okay, so we kind of wanted to understand this transition a little bit better. There was some work that was done by Fred McIntosh, and the idea was that if you have isotropic rigidity percolation happening at ZC, this critical point, if you were on the other side of it, if you were floppy, okay, so you didn't have any bonds that were stretched yet, if you were floppy, as long as you increase the strain, eventually you stretch the bonds and you can go from a floppy to a rigid state, okay? And we wanted to understand what's controlling this transition. Is there another critical point besides the critical point associated with isotropic rigidity percolation that's controlling the phase behavior? Yeah. Yep. You're always stretching. No matter what you do, you're always stretching. Yeah, so in the experiment, the way we control the density is by looking at different depths. You get it automatically. So you just look deep in the tissue, the bond occupation probability is high. You look near the surface, it's low. And so just by looking as a function of depth, you can do your entire measurement. Yeah, so under compression, you don't get much change in the modulus for the deep zone, but you get a huge change for the surface. Okay, I'll show you some data for that. Okay, so let me take you through a little foray into theory land that I did, or at least it was an idea. And the idea is the following. So the question is whether or not there's a formalism for dealing with all of this kind of shifting of the phase transitions. And the answer is yes, and it's called crossover scaling. If people have been exposed to renormalization group approaches, this is kind of equivalent to what happens when you take a Heisenberg system where the spins can point in any direction, and you break the symmetry of the Hamiltonian to an Ising model, which can only point up or down. And so what you do is, in that system, in theory land, in your little Hamiltonian space, you're going from a critical point associated with the Heisenberg magnet to another critical point associated with the Ising magnet. And there's a very well worked out formalism over the last 50 years that allows us to figure out how to essentially show that we're getting crossover scaling from a regime that's controlled by this critical point to another one that's controlled by this critical point. Okay, and so I want to kind of explore that idea a little bit. And this is an area that's still under active research, both in the hard condensed matter and in the soft condensed matter communities, when we're thinking about how to describe systems ranging from quantum materials to swarms of midges. All right, and this is all beautiful work here, showing that depending on which terms, in this bottom case, which terms you incorporate into your midge swarm dynamics, you are controlled by different critical points. And these arrows are showing you how you move from one critical point to another as you include or don't include various terms in the Hamiltonian. Okay, so it's a little bit, I'm being a little bit vague, but it's a pretty well worked out formalism. So let me show you how we go about doing this. Okay, so we're gonna look at crossover scaling of rigidity percolation in anisotropic networks. And the idea that we have is that, well, you know, when you're pulling the fibers, when you're shearing them, you're pulling them along one direction, you're aligning them. Okay, so what happens when the networks originally are aligned, right? If I have a network that is very flimsy and I pull on it, essentially what I'm saying is that I'm bound to get a shear modulus because all I need to do is stretch one fiber out all the way, and I'm gonna get it to be rigid. And so maybe the thing that's controlling the rigidity percolation under a strain is just the alignment of the fibers. If I pre-aligned the fibers, then I wouldn't have to go to as high of a volume fraction before I got a modulus. Is it clear? Okay. So in the limit, if I have a very flimsy network, okay, that would not be, that's, you know, well over here, if I start pulling it, okay, one of those fibers is eventually going to stretch. Okay, so that's like saying, what if I had rigidity percolation in a tissue where I bias the fibers to already be aligned along one direction? I'm going to reach the rigidity percolation at a lower value of P, okay, because it doesn't take very long to reach that over there. Okay, so this was the idea that we had. And so the first thing that we wanted to look at, again, was to try to think about these systems on a lattice. So again, this is the idea behind percolation, but it's not until you strain this that you get the system. So over here, if you don't penalize the bond bending, then this is considered to be a floppy network. Okay, But if you have enough of these constraints, then the system becomes rigid and can transfer forces. Okay, so that's the idea behind this very theoretical, bare bones rigidity percolation model. Okay, and what Fred had shown was that when you look at a triangular lattice, okay, again, you can get a bond occupation at around 66%, which makes the system have a rigid cluster. And as you increase the bonds further, you essentially just make the tissue or the network more and more rigid. Okay, so this is what it looks like with 65%. This is what it looks like with 70%. And the red is telling you how much stress is being carried by the various bonds. All right, and if you want to then investigate what happens under stretching, you might wrap this around a torus. So you make continuous boundary conditions on both sides. You increase the radius of the torus. That stretches the network, which is what William has done here. And then you can essentially figure out, like minimize the energy for the orientations of the bonds and calculate the derivative of that in order to get the quantity that you care about, okay, which is the modulus. So these system sizes ranged from 30 by 30 to 500 by 500 nodes. We needed to do like 10 million realizations, so it was very useful to do this on GPUs. And Will, who was an undergraduate working in my lab, really figured out how to like speed up the simulations by like a factor of 1 million in order to get this stuff to work. He was a computer scientist, so he knows how to do these things. But that was really important in order to be able to do this in a timely fashion, okay? So now each of these moduli, okay, so each modulus in an isotropic system is going to vanish like a power law, what I'm showing you here, at the rigidity phase transition, okay? So it's close to 66%, but not exactly. And the idea is that the point where it vanishes is going to be your critical point for P, where you get the transition. All right, so that's what happens normally. But the problem is that if you don't have an infinite lattice, okay, then getting the power law for that curve is really difficult. And so what you have to do is you have to ask yourself, well, how can I do experiments with different lattice sizes? Which result should I trust? How do I extrapolate to the infinite system? And this is a technique called finite size scaling that is now well worked out. And that's part of the homework that I gave you, or the problem that I gave you, was to figure out how to do finite size scaling, so that by actually looking at networks that were small all the way to networks that were the biggest that you could get, by seeing how the properties of this curve change when you change the size of the network, you can extrapolate to an infinite system. All right, so let me show you how Will did this, again, for the isotropic case. So if you take a look at these curves for the moduli as a function of bond occupation probability, what you see is that you have a different bond occupation probability threshold depending on the size of the network that you're simulating. And for each of these networks, if you have fewer nodes, then your probability distribution is wider. In other words, you get more noise in your values for P. Okay, so these Gaussians are telling you that for 150, I have a fairly nicely peaked Gaussian, but if I'm at 30, it's very, very broad. All right, and what you can do then is look at or develop or posit a invariant scaling variable, okay, which has not just delta P, but also a length dependence with some power law. And once you do that, then you can take this raw data and you can scale it so that now all of those Gaussians go on top of each other. Okay, that tells you that you really understand how things are changing with network size. You can also do this for the data for the modulus as a function of P and get them all to collapse onto a single universal curve. And when you take L to infinity, that's going to tell you how the properties of your network are going to change, how your critical threshold is going to change. Is it clear? So that's the problem set that I gave you. I gave you Will's data and some hints about how to then take the curves that you have here and try to collapse them onto a single universal curve. All right? Okay, so we can do that for the isotropic system, but as we said, our goal is to look at these networks that are anisotropic, and this anisotropy in networks is important for all sorts of things, not just cartilage, but also for the cytoskeleton. It's important for actin assembly. And the idea is to now, instead of looking at isotropic networks, we're going to bias the probability that we're going to lay down bonds along the horizontal direction by some value r. And so when we do that for r equals 2, this is the kind of network that we generate. You can see many more horizontal fibers. The other thing that we pointed out, or that we noticed, is that just doing the modulus doesn't really, is not really a great way to do it, because if you think about the modulus, it's pulling things along the 45-degree angle. But the hexagonal lattice has 60-degree angles, okay? So we're going to do it slightly differently. We're going to look, instead of just the shear modulus, we're going to look at all the components of the stress tensor, and we're going to align things along the horizontal direction and look at the change in the modulus for extending along that direction, all right? And see how that changes compared to the other ones. So in particular, we're going to keep track, by stretching the network, we can keep track of CXXXX. That's the direction that the bonds are aligned along. CYYYY. This is the shear, CXYXY. And from isotropic expansion and these two, we can also back out CXXYY. Okay, so this gives us, for 2 dimensions, in an anisotropic system, you have 4 components of the moduli of the stress tensor, and we can get all of these from these kinds of deformations. All right? Okay, and when we do that, we get exactly what Leo said, which is that when I start increasing the bond anisotropy, okay, when I start aligning things along the x direction, this curve shifts over to the left. And if I go further, it shifts over more, right? And in fact, you can look at the alignment, at the bond occupation probability threshold as a function of alignment strength. On this side, it's floppy. On this side, it's rigid for the CXXXX, right? And you can see that this is moving more and more to the left, right? Which makes sense. What we also found was that there was a second threshold for the other 3 components of the stress tensor. And so instead of just being a single critical point, this critical point ends up being a multi-critical point that we can split up. Okay, so over here in this regime, you've rigidly percolated along the XXXX direction, but it's not until you get to here, crossing the green threshold, that you start to have networks that can rigidly percolate the other components of the stress tensor. And that's super interesting, because in many tissues, cells can control the alignment of the fiber. They can control, the various ways that they're laying down that collagen network in order to generate different mechanical properties, right? And so we think that this is really important now, not necessarily for the linear properties, which are controlled by the isotropic rigidity percolation critical point, but for nonlinear properties. Okay, that's where the fiber alignment that I was telling you about, that the guy discovered 100 years ago, Benioff, right? That's where that stuff is going to come into play. Yes. Okay, and this now becomes a multi-critical point that splits up. And by the way, this is just for 2D. In like 3D, there are many more components of the stress tensor that you can bias, depending on how you lay down your fiber. And so there's a huge phase space in which you can start to organize these rigidity thresholds along different modes of deformation. And that's super interesting, right? Like that is a completely different way to think about how these sort of tissues are generating their structure and why they're generating their structure for. Okay, so if you take a look, here again is the finite size scaling for the isotropic network. And now you can do the same thing for the aligned network. And here is what you get for the aligned network. And you need another sort of scaling variable to figure out how the finite size scaling works for the aligned networks as well. Okay, but you can do that. And so here are the different components. Now you are putting in the various finite size scaling effects for the CXXXX component of the modulus, and then the other three components are shown in green. They end up being the same. Okay, and these, I'll just show you what they look like. They have different shapes. They're most likely different power laws. The fact that they're different power laws tells you that they are different universality classes. Okay, the power law determines the universality class of the network. You have a different power law, and you have a different universality class. So you're really moving the position, and you're changing the nature of the mechanical phase transition by doing these kinds of games. Yes? I don't know. R equals 1 is isotropic. So that's where all the bond orientations are uniform. If you did the R's for the other components of the triangular lattice, right, then that would be kind of like the negative. Okay, so it's, yeah.
Just wondering what procedure did you guys follow to do finite size scaling?
Yeah. So we can, that's pretty in the weeds, but you can see that this has a different exponent for the finite size scaling than the other one does. And so the idea is the same. You do the anisotropy calculation for different size networks, and you try to figure out how that works as you extrapolate to infinite systems, right? What that power law looks like. But it is tricky, and we're still not 100% sure that we understand what that power law is, okay, whether or not the delta P power law is the same, okay, and so, or different. And so I'm not quoting it here. Like, I don't actually know. Okay? Leo. These are all simulations. Well, we're getting it, okay. Almost there, okay? Okay, so let me again, like, say what we think is happening. We think that you start off with an isotropic network. That's the one over here. You start stretching the network. Maybe it becomes aligned. Now it's controlled by a rigidity percolation over here with a different threshold, possibly a different universality class. We're not sure yet. And the idea is that if you start off rigid, you can become very rigid. In other words, you strain stiffen as you do this. If you start off floppy, then you become rigid. Okay, so you undergo this transition. In the cartilage, we're always rigid. There's always a finite modulus, no matter what the concentration is, otherwise the tissue would fall apart. So we really think that this is the kind of strain stiffening behavior that we're going to get. And so let me show you some evidence for this. So again, we talked about how when you start doing things like straining the system, you end up modifying the behavior of the region near the surface. That part, that's the part that's closer to the rigidity percolation threshold. And indeed, when I apply a shear strain that is larger and larger, I can make this part stiffer and stiffer and stiffer. And the deep zone doesn't really change by much, okay? Now, let me tell you what we're showing here. This is data that's kind of hot off the press. So let me see if I can describe it. When I do a stress versus strain curve for the cartilage, okay, so here's a stress, here's a strain. And if you have no stress, right, if you're near equilibrium, then this thing has a fairly shallow modulus. It looks like this. The slope here tells you what the modulus is. If I start straining very high, then this curve goes up. Okay? And this curve will go, will diverge in that direction. All right? So that's the nonlinear response of the cartilage. If I now take, so there are different ways that I can characterize this curve. One is to take the modulus, which is just the derivative of the stress divided by the value of the strain at each point. But I can also do this. I can take the slope here. That's the modulus. That's the normal modulus. But I could do the slope here. Okay, this is called the tangent modulus. It's just looking at the derivative of the stress versus strain curve. Okay, and I think if you're thinking about stretch networks, you don't really care about this point. What you care about is stretching relative to some like point where you begin to stretch. So I think it's the tangent modulus that's most important. So what I'm going to plot for you now is the tangent modulus. This is the tangent modulus as a function of strain. And when I look deep, when I look near the surface, I might get a curve that looks like this blue one. Okay, so the modulus has a certain value at low strains, and then it increases as I get to high strains. Okay, if I do this a little bit lower in the tissue, okay, the collagen concentration is increasing, okay, then I get this curve. Then I get that curve as I keep going lower, and that curve, and that curve. So all I'm doing is I'm just doing this experiment, measuring it at different points in the tissue. This is real experimental data. And when I get to the deep zone, now I'm doing these curves. All right? Those are all the curves. Now what this crossover scaling does is it tells you that you should be able to collapse all of those curves onto a single universal curve. And what it tells you is that the region of the curves near this part, where you haven't strained very much, should have one divergence associated with rigidity percolation. And the region of the curves over here on this end should have a different, they should be able to collapse at some value, okay, of a parameter, okay? And so this is what it looks like over here. So all of these curves can be collapsed onto a single universal curve, where this range of the curve tells you about the isotropic part, and this divergence is a new divergence that tells you about the shear stiffening part of the behavior, okay? And what's nice about these scalings, the power of them, is that they show you how you combine parameters like the strain and the concentration together in order to determine the properties of the tissue. Okay, to collapse onto this curve, I had to take curves with different values of the concentration. And depending on the strain, I got them to collapse. So that's telling you what's the magical combination of strain and concentration that's going to overlay these things onto one universal curve. Okay, and that's the power of crossover scaling. Right? So it teaches us something about the system. And then by using it in order to sort of manipulate our phase transitions, we can start to think about how to really control the emergent properties and functions of our tissue. Right? So that's kind of the basic message that I want to say. Here's the same curve now without it abutting the edge of the graph. Okay, so you can see the scaling. And this is, you know, over 3 orders of magnitude, okay, from here all the way down to here. Okay, so that's 3 orders of magnitude. It's quite an extensive range. Yeah. Yeah. I will take you through an example that we've worked out much better tomorrow or Thursday when I do my last lecture. And I'll do that for the colloidal suspensions, okay? But over here, I just want to kind of point out that you can get this universal scaling and something about the power that it gives you, which is that you can now understand where that boundary is going to be depending on the concentration and the strain based on this universal curve. All right? Yes. Yes, the fibers are getting stretched and the tangent modulus is increasing like a bat out of hell. That's still a term that people use. Yeah. It's just technical stuff. Nothing. There's no gap. We can get this all across the tissue. Yeah. All right. So, so I hope that I've sort of given you at least a taste of how we're thinking about this biological tissue and how we're trying to think about the mechanical phase transitions that it goes through in order to sort of position things like diseases of cartilage. We're also trying to think about morphogenesis of cartilage. So it turns out that we can make collagen scaffolds that are very dense in collagen, but the problem, and therefore are stiff enough. But the problem with that is that it's very difficult to get nutrients in through those stiff collagen gels. So one idea that we have is maybe we print the tissue just above the rigidity percolation threshold, and then nutrients can come in and the cells can drive the network above the critical threshold, right? Or drive the cells further from the critical threshold and increase the modulus of the material. And so this is really changing the way that we're understanding how diseases of cartilage happen. It's also changing the way that we're going to think about printing biological tissues. And that's the sort of strength of this approach. And then in terms of these other tissues that we've talked about at the very beginning, again, this idea of pre-aligning fibers in order to control the function of these tissues, especially in the nonlinear regime, becomes super interesting, right? Like, how do I put these things together in order to get these emergent functions? And then how do I, once I have that, how do I hierarchically, put these together in order to get the behaviors that you get, for example, in your intervertebral disc, right? So that's the program. And hopefully we can make progress on this and start to print not just passive collagen-like tissues, but actually active, mechanically active tissues that can withstand 60 plus years of loading. That's the dream. And hopefully this kind of framework gives us a way to get there. Okay, so I want to just acknowledge the tremendous crew of people that I've worked with in order to, to get here. And so these are all the faculty, including Larry, who's been we've been collaborating with for 18 years on this stuff. Bulbul and Jim, who have really been instrumental in teaching us about all the crossover scaling, both in this material and in shear thickening. We'll talk about that tomorrow. And Mumita, who pointed out that something like this mechanical phase transition could be happening in cartilage. This is my crew of students that have worked on this problem, and these are the students from other teams that I've had the privilege of working with. And with that, I'll thank you for your time and take any other questions that you have. Should I put this up? Put this up. We've had a lot of questions throughout. Are there any others? Yeah.
I know you don't. But maybe you'll be happy to answer it without a lot of detail. So in terms of the scaling collapse for the collagen, I know you don't want to get into the weeds yet because it's work in progress on what the scaling function looks at.
Yeah.
Hear about what variables are going into scaling.
Yeah. I mean, what you care about is that this thing is going to look like some function, okay? And that function is going to have two singularities. One singularity is going to be associated with the isotropic phase. So that's going to be your rho minus rho c to some power, okay? And the other part is going to be some universal function that I showed you what it, I didn't show you what it looks like. I showed you what it looks like. Okay, that function, okay, is going to be, and it's going to be a function of some variables like the strain and the distance of this from the isotropy and R, okay? And the idea is that this part is going to take care of the divergence at low strain, okay? So I guess not R because we're doing strain. It's going to take care of the divergence at low strain. Okay, so I can pop this over to the other side. And then the rest that remains, okay, that's what is the y-axis here. The rest that remains is going to be a function that is constant at low strain, and then going to have a new singularity at high strains. Okay, so that's what these universal functions tend to look like. And depending on how this blows up, if it has the same power as the part for the isotropic system, then it's in the same universality class. If it has a different power, then it's a different universality class. And that tells you that there's different physics involved in setting that power law scaling. All right, and that's powerful. And the fact that you can do this combination of parameters, that's really telling you something deep about the behavior of the composite network, right? It's not just telling you, hey, you know, you're straining the fibers. It's telling you, no, for this concentration, this is how much strain you need. And this is how you get closer to that threshold of a fully rigid system where the fibers are being stretched. Leo, do you,
Minor question. Well, one minor. There's an exponent there, probably.
yeah, there's other exponents in there. It's just a function of these variables. Sorry. No. I haven't told you what the relationship between these are. Strain. So in other words, you take the tissue when it's unstrained, and then you're straining each layer gets strained by a different amount because it has a different modulus. So for each layer, you define the amount that it's been strained. Okay, so that's going to vary with depth too. That's why when you took a look at the tangent modulus, the function of strain, this curve right over here diverges at large strain. That's near the surface. But the regions that are deep have a higher tangent modulus to begin with, and they diverge at lower strains. Okay, and so that's why they're over here. This is deep, and this is surface.
Somehow then it says.
Yeah.
The expression for C, but C is also entering in the gamma of C.
Yeah, so the part over here, okay, is the scaling in this, like at low strain. That's how much these are scaling by. Okay, and I'm still getting all the data, but there should be something like as a function of concentration, right? You should have some power law of the modulus as a function of the concentration at low strain. And then you'll have a different power law associated with the stretching of these curves in this direction. Okay, so that's going to go into setting these power laws.
Yeah. On one hand, that's a probe of the network. On the other hand, with stress, but on the other hand, stress itself changing the network. Very nonlinear way.
Yeah, you got it. It's highly nonlinear. But the idea is that you can capture that nonlinearity. If you get this kind of scaling, it's telling you that there's some sort of universal features that are capturing that nonlinearity. And, you know, that's what we're trying to get at, right? And it's you know, anyway, it's a work in progress, but I think the overall sort of gestalt of trying to think about these systems near mechanical phase transitions is a very powerful idea, and one that really hasn't been explored very deeply in these extracellular networks. And I think that cartilage is a particularly nice one, but there are many others. And it's a really, I think, fruitful and interesting area to investigate. You had one more? Other questions? We're a little bit over time. So why don't we save the rest for afterwards, and thank you again.